I have you loud and clear.
Hello.
Welcome. Science.
And that is to say,
physics,
Medicine.
nature,
or
Space.
Time.
The brain. Life.
The universe.
Hello, this is The Naked Scientist, the show that brings you the latest in science, technology and medicine. I'm Katie Haylor.
And I'm Chris Smith. This week, testing, tracing and monitoring. How does a COVID test actually work? Do antibodies to COVID mean immunity? And the tech to monitor COVID symptoms from home.
Plus in the news, as lockdowns go local, how are they doing it down under? Why planting trees to capture carbon might not necessarily be the best move, and the sparrows changing their tune.
The Naked Scientist podcast is powered by ukfast.co.uk.
First up this week, the city of Leicester has been making the headlines. Now, unlike the rest of the UK that celebrated Super Saturday as the lockdown eased, there was no grand reopening for the pubs in Leicester. Schools have been restricted to children of essential workers again, and non-essential shops have had to pull down the shutters because, owing to a surge in coronavirus cases, the lockdown has been extended there.
Nobody seems to be able to put their finger on precisely why Leicester has come top of the league for coronavirus cases this last month. The Department for Health and Social Care said there were multiple factors causing the spike and that there were no, quote, care homes, hospital settings or industrial processes that would immediately explain the apparent rise in new diagnoses.
But scientists say that infections might reflect social inequalities. In places where there are more white-collar jobs, employees can work from home and they can isolate from others. In more deprived areas, on the other hand, people are more likely to have to go to work and to use public transport to do that, and that raises their risk of becoming infected. Cambridge University's Freya Jeffcott studies disease outbreaks. So what does she make of the longer lockdown approach that's being used to control the outbreak in Leicester?
I think in general terms, we're already sort of on the right track. It's really about sort of empowering local health authorities now, when we do see sort of increased transmission in specific areas, to act in a way that they know is locally appropriate and also locally effective to contain it, because the way outbreaks spread is obviously determined a lot by the population it's spreading in, the sort of density of the housing arrangement, the kind of industries they rely on for the economy there.
Do we actually know what is the right way forward, though, in terms of when you have these sorts of combinations of factors, what the right thing to do is? Or is it going to be a learning exercise and different cities with different formats, different population groups are going to have to learn the hard way and then they'll know for next time?
Absolutely, especially with such an unfamiliar and new virus. There is going to be a lot of trial and error, a lot of trying to learn from each other and also trying to look locally to see what's going to help. There is not going to be a one-size-fits-all, and I think that the fact that nothing is certain should be a bit reassuring in that there is discussion and research, and that's really what's required at present.
Given it is summer, and we know that viruses spread much less well in summertime when more people are out and about, we're also in the immediate aftermath of a lockdown, when arguably the amount of virus circulating should be extremely low. Is it not rather worrying that we've got Leicester happening right now?
It's obviously concerning. I'm not sure whether we should see it as entirely surprising. There are a few features of a city like Leicester that would leave it a little more susceptible to kind of ongoing transmission. What encourages me is that it was detected and that there is this discussion now of maybe some kind of local interventions that could come into control. There is something to be said for the fact that it is summer and there does seem to be less transmission that occurs when you're in sort of open spaces. So maybe there is some kind of happier middle ground of opening up that is possible with something that sort of leverages the fact that we can be outside right now and not be rained on or chilly.
Freya, we've now got plans being laid for opening up the hospitality industry more, opening up air bridges to other European countries. What are your feelings on the direction of travel? Is it too much too soon, or do you think this is about right?
I think it's important in answering this question that I make it clear that my background is very much coming from the sort of epidemiology, anthropology side. I'm not an economist, I'm not a public policy maker as a rule. But from my perspective, it is worrying. I'm sure that there are ways that we can support hospitality industries to open up in a way that sort of mitigates the risk. And so I'm hopeful there. But the issues around international spread do concern me. Not just the idea of importing cases into Britain if we get transmission even lower, which could trigger new ongoing transmission, but also the fact that we might be exporting cases to vulnerable places. We've not only got to think about risks to our country, but the role we might play in sort of proliferating this outbreak globally too.
Freya Jeffcott. And speaking of the international situation, in Melbourne, Australia's second biggest city in the state of Victoria, the government there have imposed what appears to be a much tougher local lockdown than the one in Leicester.
The finger in Melbourne is being pointed securely at security guards. Now, they've been blamed for a spike in cases because they've allegedly been catching coronavirus by having sex with international travellers who are quarantined in a hotel in the city. Here's Monash University's Philip Rousseau.
The past 10, 11 days, we've had an increase of daily new cases of COVID infection. The evidence is showing us that they're confined to a certain few suburbs in Melbourne. Some genomic testing has demonstrated that most likely they are due to a breakdown in infection prevention in the quarantine hotels that we put returning international travellers in.
What do you mean by that? There are people who were admitted to those venues and they've unfortunately spread it beyond them.
Returning international travellers are quarantined here for 2 weeks on arrival, and they've been put up in hotels staffed by security guards who have been employed to ensure the travellers remain inside the hotel. But it appears as though the security staff haven't been following the infection control guidelines. The security guards have become infected and then subsequently taken it back to their areas of residence and into the community.
How many cases has Melbourne got now, and over what sort of timeline?
About 2 weeks ago, we were having 0 cases on a daily basis, and at the moment we're having in excess of around about 60 to 70 new cases per day, which possibly doesn't sound like a lot, but then compared to the other states in Australia who are having 0 to 1 or 2 cases on a daily basis for a number of weeks now, it certainly stands out.
And what has been the reaction of the government to this?
The Victorian government has been very proactive in responding to this, and as of midnight tonight Melbourne time, there are 10 suburbs around Melbourne who are going back to stage 3 restrictions. People in those 10 suburbs will only be allowed out of their houses for 4 reasons: essential shopping for food and supplies, caregiving, to exercise, and to study or work if they can't do that from home.
And how long is this going to go on for?
So at this stage, the government is saying that this is going to continue on till at least the 29th of July, so about 4 weeks. But it's possible that if we continue to get an increase in new cases, that time frame may be extended. We are doing extensive testing in those suburbs, but also we have free testing across Melbourne. The likelihood is if we start to see cases outside of those areas, then the number of suburbs that go into that stage 3 restrictions might actually be extended.
It's a stark reminder, isn't it? Because Australia's had a very good track record of dealing with COVID. The testing was very fast off the board, done at enormous scale. It's an outlier in that respect, and it just serves as a reminder that no one is above COVID.
And that's the message we're trying to get to the community here. On an international scale, we've done reasonably well. But the message we're trying to get across is that it's not over. This is a marathon, it's not a sprint. We're somewhere in the marathon, we actually don't know where we are. We might not even be halfway at this stage. And so what we're doing now, it's more than likely that we'll continue to see these clusters, these little outbreaks here and there, possibly up for the next 12 to 18 months. We're confident, though, that the amount of testing that we're doing and our public health response should be able to limit the spread of the infection to a large degree.
So, both some good news, but also some bad news. A sort of sombre message there. Monash University's Director of Nursing Research, Philip Rousseau. Meanwhile, UK government sources are saying that Leicester and also Melbourne-style shutdowns might be just days away for other places if us Brits continue to flock to bars off the back of the Super Saturday lifting of restrictions in the lockdown.
Over the past decade, planting trees has become an option backed by governments to capture carbon dioxide in the fight against climate change. But a new study shows that this can leave the planet worse off than it was before. Researchers have analysed the effects of a long-running campaign in Chile to subsidise tree planting. And as Phil Sampson heard from Robert Heilmeyer, the resulting plantations weren't good news.
In many cases, those new plantations were encroaching upon native forests and shrublands, and so that conversion of native forests to plantations really led to a reduction in biodiversity across the landscape and an undermining of the carbon benefits that we would hope we would get.
Really? So in this case, planting trees ended up bad for the environment?
Yeah, I mean, what it ended up highlighting is that it really depends how you go about planting those trees, right? If you're replacing some of the existing standing forests that's been growing for centuries and replacing them with kind of young plantation forests that you plan to clear within 20 years, obviously you're not going to get as much carbon in that above ground biomass that you would if you just let those forests standing.
Can you explain that? I'm not quite sure I understand the whole biomass concept.
Yeah, so, all vegetation is made out of carbon. And so as plants go through photosynthesis, they're storing carbon in their structure. One of the ways that people think we can, a potential contribution to addressing climate change would be to increase the amount of vegetation out there. We're kind of increasing the storage of carbon.
Basically, more tree material equals good.
Yes, that's usually the case, yeah. And so what ends up happening with this kind of plantation setting is that you have less tree material, less plant in a forest than you would within kind of a natural, dense native forest.
That's kind of perverse, though. Why were people chopping down beautiful old forests just to plant a new single type of tree?
Yeah, I mean, the initial intent of the policy was largely to build kind of a domestic timber sector. This was implemented under the Pinochet regime in the 1970s, and they were really looking for ways to kind of drive economic growth. But at the same time, there were kind of environmental concerns in this kind of central and southern parts of Chile. There were large areas that had been degraded through really aggressive agricultural practices. And so the government felt that if they encouraged people to reforest those landscapes, they would reduce the rates of erosion and help kind of reclaim some of these lands that were really marginal and not being put to use. The problem with it was that although they initially had a policy in place that said you can't convert native forests to plantations and get this subsidy, they were really poorly enforced. And so you had a lot of cases where you were actually getting the kind of conversion of a native forest to agriculture for a year, maybe a pasture, and then people would apply for these subsidies and convert it to plantation forests.
And then if the point was for timber, were people just getting paid to plant trees and then chop them down again?
Exactly. So they do provide some soil erosion protection. They do provide some carbon benefits, but the main goal of those plantations is really as a feedstock for large pulp mills and timber mills that exist in Chile.
Can you then compare this to any of the big tree planting campaigns that are going on today?
We don't necessarily think that planting trees is a bad idea. It can be, and I think it will be, an important part of the solution to climate change. However, our study really cautions that you want to design the policies in a way that doesn't lose a native forest at the same time. We want to be reforesting landscapes with kind of the native species, not an unending kind of row by row monoculture of one species of trees.
It reminds me a little bit of the whole biofuels thing, where people thought, here's an environmentally friendly fuel. It just grows out of the ground. But in reality, it takes precious land to grow that biofuel, and then you're just burning it anyway.
Yeah, I think that's true. And even, in the timber sector, a growing trend right now is to use wood pellets for home heating. And I think that's especially common in Europe. And what you're finding is that in some cases, those wood pellets are associated with clearing of natural forests at a pretty large scale. I think policymakers today are making some of the same mistakes that we saw happen 40 years ago. When you look at the commitments that people are making in things like the Bonn Challenge, which is one of these kind of global reforestation campaigns, almost half, I think, of the commitments under that are to replant with plantations.
Robert Heilmeyer of the Bren School of Environmental Science and Management, and his study is in the journal Nature Sustainability.
Critics everywhere are raving about the Naked Genetics podcast.
I wish I could show you it. It's beautiful.
This is really exciting.
No, that is actually quite disgusting.
Potentially a global phenomenon.
I'm out of here and I'll be watching YouTube instead.
Come on and join me, Phil Sansom, every month as we shrink down to microscopic size and understand the quirky code inside all living things. From the science behind the headlines.
SARS and COVID have hijacked this essential protein and in the process wreak havoc in the patient.
To the obscure and unexpectedly fascinating.
It's not overly pleasant to send a diver swimming through a massive plume of whale shark poo, but you've got to make these sacrifices for science.
Subscribe to Naked Genetics on your podcast app today.
On the way, Test, Track, Trace. We're unpicking the science behind COVID testing.
Before that, though, how the immune system works and the role of our genetic makeup in causing a range of inherited immune diseases, including autoimmune conditions, allergies and increased cancer risks, have been revealed by a landmark study from researchers at Cambridge University. The team have read the complete genetic codes of over 1,000 patients with these sorts of disorders to link the genes they carry with the diseases they go on to develop. And here to explain how they did it and what it's showing them is Ken Smith from the Cambridge Institute of Therapeutic Immunology and Infectious Diseases, CITID for short. So Ken, tell us first of all, how did you do this?
We did this by studying a group of patients with defective immune systems with conditions called primary immune deficiency. And these patients are susceptible to infections, often very severe infections. About a third develop inflammatory diseases and about 10% various sorts of cancer. And this is caused by genetic problems, but in the vast majority of patients with these conditions, the genetic cause is not found. So what we did was to recruit a very large number of these patients from over 27 centres across the UK. So we ended up with more than 1,000 patients, which is about more than a third of patients with this condition in the country. And we sequenced their entire genome rather than the usual approach, which is to sequence just a small proportion of it. By doing that, we could then discover new genetic causes.
I get that you got these patients, you know they have a condition that unites them and that you read the DNA end to end. But if you read a book from end to end, you wouldn't necessarily know which chapters were the ones playing the role in the story that you're referring to. So how did you link the diseases these people had with what the genetic code was saying?
So what we did was to look for mutations or variants that were present in patients with the disease, but were not present in a large number of healthy people. So if a few of these very rare mutations were found in a gene in patients with immune deficiency and were not found in the normal population, that made us very suspicious that that gene was involved in causing immune dysfunction. What we then had to do was go to the laboratory to prove the association of that gene with immunodeficiency. What that did in this study was to allow us to find more than 5 new genetic causes of immune dysfunction, with many more, I think, still to come from the study.
Why was this missed before? It's pretty straightforward what you've done in the sense of reading people's genomes. Why had no one thought to do it the way you did it?
I think they probably thought to do it, but it's only become affordable recently. Until recently, it was impossible to sequence whole genomes at scale, as we've done. But through the National Bioresource Rare Diseases Program, it's become possible to afford to sequence patients. The second thing is the statistical approaches, headed by Ernest Turrow and others, that have allowed us to analyse such large numbers of genomes. So it's a technical advance rather than a philosophical one.
Obviously, it's very useful for patients who have one of these conditions because it means you can give them almost a more precise diagnosis. But Is it true that when nature makes a mistake in this way, sometimes some of her other inner workings are revealed? That's the kind of the old saying, isn't it? Have you learned more about how the immune system works in a healthy person by studying a less healthy person?
So we've found a number of genetic pathways involved in causing these problems with the immune system that we have evidence are involved in much more common diseases as well. And by finding that information out, we can study more about the immune system in general. What we've also done for the patients who get the diagnosis is put them in a position to plan their lives better, understand the implications of their disease for their families, which is often not known until they have a genetic diagnosis, and in many cases to allow specific treatments for the disease that wouldn't have been possible without a diagnosis.
Wonderful. Thanks for joining us to talk about it. Ken Smith from the University of Cambridge.
And finally, in the news this week, researchers in Canada have found that there's a new hit song sweeping the country, at least among the nation's white-throated sparrows. When biology professor another Ken, Otter, moved to British Columbia in Western Canada, he was surprised to discover that these small and noisy birds were singing a very different tune to their East Coast colleagues. And as the years went by, recordings sent in from citizen scientists all over Canada showed that this West Coast new song had gone viral.
What it is at the beginning of the song, the males have an introduction that has 3 whistled notes. It's a very sort of clean, pure sounding notes. And it's a very patriotic in Canada. At least the description of it is that they're supposed to be singing, "My, oh, my sweet Canada, 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 Canada." And so that ending phrase is a repeat of 3 repeating notes with a little gap in between them. So it's da-da-da, 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 like that. And the males will sing that variable number of times. That's the classic triplet ending song.
And you've actually sent me some recordings of that, so let's have a listen and hear what that sounds like, the triplet. Okay, so I can hear the Canada, Canada, Canada. So that's the classic. What's the new one?
That's the classic. The one that we found, instead of singing 3 notes, they sing 2 notes. It's kind of a long note, short note, long note, short note with not much of a space in between it. And then they end it on a long note. So it kind of goes canna, 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 Canada, like that. So it's like they're stuttering the whole way through it.
Okay, cool. Let's give that one a listen too. Okay. So the song seems to have originated in the west of Canada and now is travelling east.
It was still the common song type in the east, across the eastern range of the birds, was the triplet ending songs in the early 2000s. And between 2000 and 2019, the doublet ending songs spread across Canada and took over. It completely replaced the triplet ending song. So it's traveling across the country at quite a rapid pace, and it's completely replacing the triplet song wherever it's taken a foothold. So what was happening is our birds in British Columbia were overwintering with birds that were singing a totally different song type. And we started to look to see whether there was a possibility that the birds were singing on these wintering grounds and potentially serving as tutors for juvenile birds that bred in other populations. And that could explain how the song actually spreads from one population to another.
And so this was essentially a mixing of birds from the west of Canada with the east of Canada. They all head down and mix up and overwinter together.
That's basically what we found, is that the birds do sing on their wintering ground, and we started to see this Western dialect popping up in the eastern wintering grounds. But what's strange about this is that we're having situations where males are coming in and they're singing this new song type, the doublet ending song, and instead of converging on what's the common song type in a particular area, these few males that are settling suddenly become the template, and other males start adopting that song, and it starts to spread in the population. And so the new song type starts to take over. And it would be kind of like an Australian person coming to Cambridge and everybody suddenly adopting an Australian accent and everybody starting to sound like Australians in Cambridge. And that's essentially what we're seeing here.
So what is special about the doublet sound that has made it take off so well?
I don't know like specifically what it is about the doublet. It may just be that it's slightly different. And there's certain templates inside the bird's head that when they actually hit on that new song, it's acceptable, but it's novel. And that's what we think might be driving this, is that they're picking it up just because it sounds slightly atypical, but it's still within, the acceptable limits of what the birds recognize as an acceptable song. And so occasionally these things might pop up, and then if they're broadly adopted, then it kind of just sweeps through the population quickly.
So it's sort of the cool new tune, which is, it's not so far out that the birds will be put off by it, but it's different enough to sound cool and interesting.
Exactly. And so that's what we think might be happening, because there doesn't seem to be a big disadvantage to sing one of these new dialects if you're trying to establish yourself as a territorial male. So we think what might be happening is that females might actually like these slightly atypical songs, and that could be driving males to adopt them. But that's something that we still have to test.
That was Ken Otter, Eva Higginbotham, and some singing sparrows. Ken's paper is in the journal Current Biology, and if you'd like to find out more, all the transcripts and papers can be found on our website, nakedscientist.com. Time now for the mailbox, where we address your correspondence. And Chris, this one's for you, because Porig has emailed in to ask about oncolytic viruses. He says, have you ever discussed the topic of oncolytic viruses on your show? Do you think this could be a useful therapy? First of all, Chris, what are these oncolytic viruses he's referring to, and what kind of therapy?
Hello, Porig. Oncolytic means cancer busting. Lytic means lysis, and onco as in cancerous. And researchers are studying viruses because viruses naturally infect cells, they grow in cells, and in some cases they destroy the cells they grow in. And that means they might be a useful anti-cancer weapon. The other thing that viruses do, which has got researchers interested, is that when they go into a cell and infect it, they obviously attract the attention of the immune system. And one of the problems with cancerous tissue is that the immune system is being programmed to ignore the cancer rather than kill the cancerous cells. And sometimes the presence of the virus there, whether or not it kills the cancer cell, will excite the immune system to the extent that it can surmount some of this suppression of the immune system and can encourage the immune response to go after not just the infected cells, which have the virus in them, but other non-infected but identical cancerous cells alongside, as well as potentially other places in the body where the cancer has spread to. These are called metastases. We have looked at this on and off over the years. There are some quite exciting projects that are being done at the moment, but they're all experimental. There is no, at the moment, gold standard therapy involving a virus that will go in and treat a cancer. One of the most exciting ones we covered, I think, is way back in the early days. And a Professor Brown, she pioneered some of this stuff up in Glasgow, developed a modified herpes virus that could be put into brain cancers, which are notoriously hard to cure. And she was able to turn people who were literally months to weeks away from dying of their brain tumours into people who are still alive today. So we know this has potential, but at the moment it's still very much at an experimental stage.
And if you've been enjoying our Science Pub quiz shows that we've been running recently, we're now offering the chance to become one of our contestants on the 26th of July. Do you think you've got what it takes to be our big brain of the month? Send in an email to chris@thenakedscientist.com or get in touch via Facebook or Twitter.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
For the rest of the show, we're concentrating on COVID. Test, track and trace is an often heard mantra at the moment, so for the next 30 minutes or so, we'll be digging into each of these T's to unpick the science underneath.
First T, testing. As of early morning on the 2nd of July, nearly 8 million tests have been processed in the UK. The vast majority of these were molecular tests, so-called swab tests that can reveal if you're currently infected with coronavirus. A swab is taken from the nose and throat, and the test looks for specific genetic material that indicates that the virus is there.
Ravi Gupta is a physician at Cambridge University, and he's been working on speeding up tests like this. So, Ravi, how do these Do I Have COVID Now tests actually work?
It's based around trying to pick up or detect small amounts of the genetic material of the virus, which of course is present during an infection. The method relies really on making large numbers of copies of what's already there, because of course what's already there can be very small, and we need to make the signal very large in order to detect it. And so that's called nucleic acid amplification, and that's exactly how most tests are developed and function. So these can be fairly complex or they can be more simple, and we've been working on a simplified version that instead of taking 3 to 5 hours to run, will be able to give you a result in about 75 minutes. This particular platform is called Samba 2. The reason it's very successful in terms of the technology and how quickly we've been able to implement it is because this is a machine that was really developed for monitoring HIV in Africa and to give a robust, quick result in a very sturdy platform. And it's been tested and tried over many, many years. And so what the inventors did is to adapt it for SARS coronavirus 2, which is the virus that causes COVID-19. And so the new chemistry was developed very rapidly, and we had the opportunity to test it at Addenbrooke's Hospital in around 200 patients in a trial that showed that the test was equally as good as the existing test in the lab.
And what is this new chemistry that enables it to be so much faster?
So the new chemistry is based around the fact that traditionally we use something called polymerase chain reaction, and that basically needs temperatures to go up and down in a very precise way, and that takes time, and we have these things called cycles involved. And the new chemistry really does something somewhat similar, except performs things at the same temperature and is what we call isothermal, and that makes it much quicker and simpler to run.
How good are these tests? How worried do we need to be about false results, false negatives, false positives, stuff like that?
In the trial that we ran, where we put Samba 2 rapid test against the hospital laboratory test developed by Public Health England, our test was 98 to 99% sensitive. In other words, picked up 99 out of every 100 positive tests using the standard laboratory assay. And it didn't get anything wrong the other way in that it didn't call anything positive that wasn't actually positive. And that's very important because you don't want to be telling people they have COVID when they do not, in fact, have it.
So 98 to 99%. Is that good enough? Can it be made better? Can it be made faster?
Well, there's always a trade-off here. The reason it takes 75 minutes is because you still have to amplify the stuff that's there in the first place, and that takes time. So if you're going to develop a highly sensitive test, you need to have time for amplification to occur. If you just use a standard antigen test, which is just looking for protein that cannot be amplified, your tests are much less sensitive in general. And so there is a trade-off here and a cost to having accuracy, and that is time. And I think an hour is actually pretty good in terms of getting a test result. The biggest delays sometimes actually are getting the swab from the nose and throat and getting the sample to the machine. If we can cut that down, for example, using saliva, I think things could get even faster.
Why is speed so important when it comes to testing?
Well, speed is of the essence because when a patient comes in to hospital, we want to prevent any transmission within the hospital. And so we have to pick out those people with symptoms who may or may not have COVID. And of course, the problem is that the symptoms are very vague, ranging from a runny nose to cough to headaches to problems with your sense of smell. So it's very difficult to tease out who has COVID and who doesn't. And therefore, we need to have a rapid test that can give us that answer quickly because the clinical diagnosis is not particularly accurate. And we need to isolate infected patients rapidly to prevent spread.
Ravi, it's July at the minute, but looking ahead to winter of 2020, there's all the kind of seasonal coughs, colds, bugs that come along with that time of year. How does this relate to testing for COVID? Does winter pose a threat?
Yes, we're all somewhat concerned about winter here in the UK because, as we can see, COVID has not gone away and we are relaxing certain measures and therefore we expect the number of cases to rise. And this rise may end up coinciding with the seasonal influenza epidemic, and it's probably going to be a smaller one this year because of social distancing worldwide, but it will still probably arrive. And of course, other cold viruses that circulate in winter. And the key thing here is that we need to design tests or have tests available that can test for multiple different viruses at the same time in order to differentiate patients and to treat them appropriately in terms of isolation. Of course, the most important virus of those 3 or 4 is going to be coronavirus, even going into winter.
But we don't have those tests right now. Is that right?
So a number of individuals and companies are looking at things called multiplex tests, which are rapid tests often that can detect a number of different viruses at the same time. We know that the Samba machine is being adapted to do a number of different pathogens at the same time, but will require validation, as we did for the single test, in order to make sure that it's as good as it needs to be. And the same goes for all the commercial tests that are being developed that go onto much larger machines and can run much larger numbers of samples. Those will also need to be tested. And this is a bit of a challenge because, of course, we're going into a period where we have declining numbers of cases, and actually evaluating the tests in real patient samples becomes more difficult as the number of cases in hospitals decreases.
Ravi Gupta, thank you very much.
So that's how we can detect whether someone has the COVID virus right now. But what about the question of whether they've had the infection in the past? Well, this is where tests for antibodies come in. If there are antibodies against an infection in the blood, it shows that a person has been exposed to that infection previously. As immunologist Chris Zhu puts it, antibodies are a bit like the footprints an animal or a person leaves behind. And if you identify those footprints, you can tell what animals have been passing by in the neighbourhood. At his company, Thermogenesis, he's invented a test kit that people can use at home for this.
The test is very simple. It's detecting humans' immune response against the virus. It takes 1 droplet of blood, and we can read the result in about 2 to 3 minutes. The test will release to be more than 90% accurate.
But that still means that you're potentially getting it wrong 10% of the time, doesn't it?
Yes, each test has its limitation. There are many different factors could be affecting the performance of a test, such as viral mutations. If the virus mutated, then certainly it will not be recognized by the antibody test. Also, the blood chemistry might interfere with the performance of the test.
So no test is perfect. That was Chris Zhu. And Chris sent me some samples of his test so that we could try it against another kind of antibody test, this time one that could be run in a laboratory to test large numbers of patients quickly and accurately. It's been developed by immunologists at Addenbrooke's Hospital in Cambridge, and I went to see doctors Sara Lear and Anita Chandra, and I asked them to tell me how their test works and also to put Chris's test to the test using Anita's own antibodies. But first, Anita answered this crucial question.
An antibody is a protein that circulates in the blood or can be in various tissues of the body, that its job is to help fight various forms of infections, be they bacteria, viruses or parasites. There are many different forms, what we call classes of antibodies, and there are lots of antibodies against different bugs. So you can have antibodies against coronavirus, mumps, measles.
Where do they come from?
We have white cells that circulate in our blood and are made in our bone marrow that have the capacity to produce what we call IgM antibodies. So these are the first type of antibodies that are our first line of defense. And they have the capacity to recognize millions of different organisms. On the first exposure to, for example, a virus like coronavirus, we would make an IgM antibody. And then at a later stage, we would make a different type of antibody called IgG antibody.
Why make 2 different types?
The IgM is produced immediately. It has different effects with other cells of the immune system. The IgG comes along later. It circulates in the blood. It has a long half-life, up to 3 weeks, and it can cross the placenta. And it's the IgG antibodies that we measure when we look at vaccine responses, for example.
And Sara, if you make 2 different types of antibody at 2 different times, does that mean you could use that as the basis of a test for whether someone's got something right now or had something previously?
Yeah, the IgM comes first. It's pretty quick. Within 24 hours, it starts being built up, peaks at about 3 days, and then the IgG kicks in after that. So if you do a test right at the beginning, you can measure the IgM, and you can tell, oh, that's more likely to be an acute infection. And then if it's not an IgM but an IgG, you can say, well, that's it. This has happened sometime in the past, and it's not recent.
that's one of the things you've done here at Addenbrooke's. You've managed to make a test that actually looks for the antibodies against this new coronavirus. How does it work?
So all these tests work with the same principle, that we need to get the bits of the virus that we're looking for, and we have to fix them somewhere and expose them to the antibody in the patient's blood. And then we have to be able to see that antibody protein complex. So in this case, we took the viral proteins and we stuck them onto tiny microbeads. And then we incubate that with the patient's serum. They stick together if the antibodies are there, shine lights at it. And if you've got an antibody-antigen complex there, then it will shine up and give you a light measurement.
And it glows only if the antibody's stuck on.
Exactly.
And it works.
Yes. Yes, it does work.
Now, I got you here under slightly false pretences, Anita, because Sarah let slip that actually you're a positive control in all her experiments because you have antibodies. You have, without realising it, had coronavirus infection. So will you be a guinea pig for us?
Yes, I'm very happy to be your guinea pig.
Okay, I understand you are the lab guinea pig anyway. This is a point-of-care test which has been sent to me from a laboratory in America. And the way it works is that we need a sample of blood from your finger, and it will register whether you have antibodies of the IgG or IgM type. You need to use this alcohol-soaked swab to make it all clean.
Thank you.
You're now going to take that green thing, which has sprung-loaded, and you hold that against your finger, and it's going to put a little tiny blade into the skin, and we should make a hole in you, and some blood should start to come out. I press that.
Apologise if I cry.
Are you alive?
I'm still alive.
Use the little pipette I've got here and just suck that drop of blood up off your finger. Blimey, there's a lot coming out. Here's a nice dry swab. This is the actual test cartridge. It's about the length of your finger, and it's about the width of your finger as well, isn't it? And there is one end which is a well where you put the sample, and then there is this area about 1 centimetre long and about 2 or 3 millimetres wide. That's where we're going to see your result. So what you have to do is take about 5 drops of your blood, using the pipette, and just drop it where it says S for the sample.
Here.
Yep, so in goes the sample. Now we just have to leave it to cook.
Okay. Is this like a little pregnancy test?
Yes, it does give lines, just like a pregnancy test. There's a C there, which is the control one, which says it has worked. And then there are 2 or 1 line will come up for antibodies on there. We'll put that to one side and we'll see in a minute what the result is. Sarah, do we feel we understand how we respond to coronavirus infection?
So I think no has to be the answer to that one. There are some features of this virus that appear very different and that are unusual. The antibody test will help, I think, to pick up how many people in the environment have been exposed to it, but we need to do more work around the acute infection to see how it actually evolves.
Do we have any feeling, Anita, for once you've made a response, whether that lasts very long or does it disappear?
That's what we're still trying to ascertain in the labs at the moment. I think a lot of that data from China will be very important because they are now at least 6 months in, and they will be able to look at the patients and their convalescent samples to see how long they have antibodies and whether they are protected.
Why should the antibody disappear, though? It seems a bit strange that for some infections you have antibody and you have it for life, whereas with this one we're uncertain as to whether it will hang around for any appreciable time. Why might that be the case?
Some of the other SARS-like viruses, they've found that the antibodies only last for 2 years. I don't know why it's only 2 years in this SARS virus, whereas for some patients like the influenza virus, there are patients who are over 100 and who were exposed in 1918, and they still have evidence of antibodies to the influenza virus. So for that, I'll have to raise my hands and say, I don't know.
What might the implications for a vaccine be then, Sarah?
Vaccines work in 2 ways, and the antibody response is only part of that. There's also a cellular response, which forms a really important part of how you clear viruses from the system.
Because researchers in Sweden this week have said that there are people knocking around who don't have appreciable antibodies, but they are sure as hell immune to this new virus. How might that be then?
When your body comes across a new virus, T cells come along and they recognise those proteins exposed on the cell surface as being foreign, and that can induce a cellular response, which means that the cell that's infected gets destroyed. And the cells will be able to recognise that combination again in the future and will then protect you for longer term.
So you can have low levels of antibody, but because you've got these T cells that recognise a virally infected cell, that's going to give you protection as well. So it's not just a one-prong, it's a two-prong response.
Yes, and there are other cells and neutrophils. When exposed to coronavirus, they produce these little nets that capture them and eat them. The immune system is very complicated, which is why it's very, very interesting to study.
Well, look, I think on that note, it's moment of truth time, isn't it? Should we have a look and see? So here is your result, Anita, and I see one very strong red line against the control, showing that the test has worked. But come on, immunologist, is there anything else on there?
I think there's a faint line on the IgG.
Yes.
It's really
very
faint.
Okay, with the eye of faith, there might be a very faint line where the IgG antibodies are. Have you had it, Sarah?
I haven't had the test.
Because you share an office with this woman.
So I'm safe, right?
Well, not initially, not potentially.
I haven't done it because I haven't been ill. But...
Out of curiosity? You haven't checked
Do you want to do a test?
I don't trust that.
Don't like your test.
I don't like that.
It's not as good as mine.
No, I believe you haven't tested. No, I just haven't got round to it.
Shall we make her do one of these?
I think we should.
So we did validate Chris Zhu's point of care test. It does appear to work and agree. Anita Chandra, who you heard there, does have antibodies against COVID. And thanks very much to Sarah Lear, who talked us through how she helped to develop it. And that test was developed as the brainchild of clinical scientist Rainer Doffinger, who works at Addenbrooke's as well.
Now, if you're in hospital, recovering at home, or you work in a high-risk environment like a hospital or a care home, regular and frequent health monitoring can provide valuable information about how you're doing regarding COVID.
Indeed, COVID monitoring isn't just limited to the indoors. This week we heard news, for instance, that monitoring sewage for the COVID genetic code could pick up infection spikes up to 10 days earlier than medical-based tests.
But back in 2019, Northwestern University's John Rogers told us about sensor technology he's developing that can non-invasively collect information such as heart rhythm, body temperature and blood oxygenation. At the time, he had premature babies in mind, but now he and his colleagues have redeployed the technology to help combat COVID. And in hospitals and homes around Chicago are 100 of these patches, which sit at the bottom of the neck and collect valuable health data about the wearer. John, how do these patches work?
Well, they're wearable devices, in the conventional sense of the term, but they're very unusual in their form factor. They're very thin, soft, and flexible. They mount on the body just like a Band-Aid, and when positioned kind of at the base of the neck, they operate as a wireless digital stethoscope is the way that you can think about it. And it's a device that continuously talks and streams data to the cell phone. And from those kinds of measurements, you can determine sort of respiratory behaviors. And that's important because COVID-19 is essentially a respiratory disease. And so you can measure all sorts of body sounds just like you would with a stethoscope. So you can hear the lub-dub sound of the heart. You can also listen to air rushing into the lungs. And during expiration, you can count coughs. You can measure intensity and sounds of coughs. We also have a temperature sensor embedded in the device, so we can pick up early signs of a fever as well.
How is it powered?
There's a tiny battery in the device, and it operates the sensor itself. There's an onboard memory module, so we can store data locally. There's also a Bluetooth radio that allows that kind of continuous streaming mode to send data to a cell phone or a tablet.
How does this monitoring compare to if you had a human do it?
Well, it's measuring these quantities continuously all the time, and it's measuring parameters that would be difficult for a human from a practical standpoint to keep track of. It's doing heart rate essentially, cardiac sounds is kind of the basis of the measurement, respiratory rate variability, core body temperature. We also have a module that allows for measurements of blood oxygen levels, and so I assume if you are a super diligent individual and you have a health diary, you could sort of track some of these parameters, but I don't think that's really practical. And in many cases, what we've seen in COVID patients that have been wearing these devices is some of the key symptoms are very transitory in a timing sense. And so there are spikes in heart rate, for example. And so if you're not tracking and monitoring these parameters continuously, you sort of miss some of the key signs of the symptoms of the disease.
Is the idea that these would be used in hospitals, in the community, or a bit of both?
A bit of both. So we have about 100 devices deployed in various areas of the medical complex here in Chicago. We were approached by physicians, doctors, pulmonologists, respiratory rehabilitation scientists, and so on in early April, and they were aware of a lot of our work in monitoring of neonatal health, and they asked us whether we could sort of adopt that technology, adapt it, and customize it to directly address the key symptoms of COVID-19, which are fever, shortness of breath, and coughing. And so that really got us started down this path. And their vision, in one that we share, is that these devices can monitor frontline healthcare workers or others who are at high risk of contracting the disease. So we can monitor very early signs of symptoms in those individuals so that they can be pulled out of the community and care can be delivered to them at the earliest possible moment. But we're also on patients at various stages of the disease itself, both in the hospital and after they're released to the home setting. So we can track the progression of the disease. We can determine exactly how their condition is evolving and how it's responding to the various novel therapies that are being addressed. So we can look at improvements. We can also look at deterioration of that health status. All the data goes to a secure cloud server, which presents the data in sort of a dashboard that physicians can inspect and base judgments on in terms of how to best treat and care for their patients.
John Rodders, thank you very much.
Now, so far in the programme, we've talked COVID testing and monitoring. So now we're moving on to that other T, which is tracking, and another T, tracing the infection. Last week, Germany launched what it claims is the world's best app for COVID contact tracing. We were expecting ours in May, but so far it's totally failed to materialise. And last week we got the lowdown on why that is from University of Strathclyde security engineer Greg Poole.
They had an app, they were testing it in the Isle of Wight, and it started to hit some teething problems around iOS devices recognising each other. And the reason for this was Apple has some technical restrictions on how Bluetooth works on their devices that they're not able to work around unless they go down Apple's approved route of contact tracing.
So what's next then? What if this app fails to materialise indefinitely? Where does digital contact tracing go from here? Well, Ramsey Farragher's with us. He's the CEO of Cambridge company Focal Point Positioning, and they specialise in navigation and positioning. Ramsey famously joined us on the show back in March, and he had a proposal at the time to use Bluetooth for COVID contact tracing. That turned out, Ramsey, to be extremely prescient to the situation. But what I don't understand, and perhaps you can clarify for me, is why is it that we can put a rover on Mars and drive it round with pinpoint accuracy and know exactly where it is, but when it comes to writing a computer program for people to run on their mobile devices, we're really struggling?
That's brilliantly put. And I think one of the reasons is because everyone's pulling in the same direction with the same priorities when we tried to put a rover on Mars. The challenge that has come about with the UK contact tracing app was fundamentally that this unknown problem occurred, which was to do with the iPhones basically being asleep when the Bluetooth signal was trying to wake them up. Instead of just turning that bit off so that wouldn't happen anymore, Apple and Google got together and created their own view of how contact tracing should be done. It prioritizes battery life and it prioritizes privacy. And that means they fundamentally change bits to do with how Bluetooth is used. And it does indeed turn out that the NHS X app was doing a better job of determining how close another device was than the Google and Apple system can do. And it's because of these priorities. And so people like me are sort of advising Apple and Google and others on changes they can make to make it a bit better. The simplest thing is to do with how often the devices try to talk to each other.
If I may, why do we have to use Bluetooth? This is a radio wave, isn't it, between two devices? And I put it to you that if you're sitting on one side of a wall and I'm sitting on the other and Bluetooth goes through the wall, the device doesn't necessarily know there's a wall there because radio sees a wall as transparent and goes straight through. Why can't we use some other way? Why can't we do it with sound? We can make a dog whistle that my dog can hear and probably my children, but I can't. Why can't we make the phones do this with sound?
We can, and people are starting to look into ultrasound for this. The reason we started with Bluetooth is because it's all set up already in the phone for them to talk to each other over Bluetooth. And you're right, the signal goes through walls and windows and glass, and that means you'll get more false positives. And so there are entities that are looking at building apps using ultrasound instead, which at the moment is a bit of a hack. So it's using the speaker and the microphone in your phone to transmit an ultrasonic chirp that you won't hear, but the phones can pick up. And you can use that to detect the proximity of two phones to within a centimetre, which is much better than Bluetooth, which is about a metre resolution.
That sounds quite promising. In Singapore, where they're conscious of the fact that not everybody has got a smartphone device and not everyone's going to want to run one of these things, and there are these headaches with how Apple have decided to inflict themselves on the world, people are using other kinds of specialist devices. They've actually issued handheld contact tracing devices that do what they do want to do without the constraints imposed by third parties. Do you think that might be a possible solution here?
Yeah, it's a really good idea. It gets around the challenges that we've seen with trying to do it directly on smartphones. And it means that vulnerable people who might not have a smartphone or might not remember to keep them charged can be given a necklace or a bracelet that does the same job and does it with a very long battery life because it's dedicated for that role. So they're a good idea, and I think we'll see them over here.
Do you think that's going to be what we end up doing then, or do you think we are going to get this mysterious app? Because Germany have done it. They reckon they've got the world's best app. They've got a problem with people wanting not to download it, but that's a different problem.
Well, independent test of their app suggests it's not quite as good as they're claiming. But I do know that the Apple Google methodology is steadily improving. So it will get better and we'll get there in the end. And people like me are trying to make everyone get there as fast as we can.
Well, we're delighted that you're on board, Ramsey. Thanks ever so much. That's Ramsey Farragher, and thanks to our other guests this week who've helped us to unravel some of the complexities of testing and tracing and monitoring for COVID-19. They were Ravi Gupta, Sara Lear, Anita Chandra, Chris Zhu, and John Rogers.
And we've just got time for Question of the Week. And Adam Murphy's been stirring things up, looking for an answer for Nirav.
How do coffee granules dissolve so well compared to soup in a pack, for example? The difference is amazing. Coffee is dissolved with minimal or no stirring.
The granules in a cup of coffee do dissolve so quickly. Just ready for that first sip. I don't like coffee. But, thankfully, University of Cambridge chemist Liliana Fruck does. And she knows how it dissolves.
Ah, that beautiful smell of coffee in the morning. And that caffeine kick that helps us to get to that early morning Zoom meeting. And making a nicely soluble instant coffee requires a bit of chemical engineering. Coffee granules we dissolved in hot water are nothing else but brewed coffee which was dehydrated. That means the water was removed either by process of spray or freeze drying. These processes result in fine powder, which is not particularly soluble. It binds the water and clumps, and it doesn't really look nice. And it also doesn't dissolve readily.
Which means something needs to be done to those very fine particles to make them dissolve so quickly. But what could that process be?
Enter granulation, a process by which fine powder particles are made larger. They agglomerate into bigger and more porous particles which have larger surface and are easier for the water to flow. There is a size which is considered very good in terms of solubility of granules, and this is usually around 250 micrometers. Now, in instant coffee, you simply have dehydrated coffee granules. You add a bit of water, and they will drink that water up and dissolve.
And then why is something like instant soup, which I do like, so different?
Soup is a bit different. It contains many different ingredients. Many ingredients with different properties, use of binders, presence of starch makes uniform granulation in soups more difficult and results in instant soup that requires usually hot water and mechanical mixing to dissolve, unlike our homogeneous and nicely granulated instant coffee.
Thanks to Liliana for giving us that solution. Join us next time when we'll be answering this question from Carol.
The government has provided more than 1 billion items of PPE to hospitals. There have been 130,000 COVID cases in hospitals, about 10,000 items of PPE per patient. Can you find out why so much?
And if you think you know the answer, get in touch. nakedscientist.com/question. You can email Chris at thenakedscientist.com, find us on Facebook, tweet at Naked Scientists, or you can join in the debate on the forum, nakedscientist.com/forum.
And that's it for this week, but do be sure to tune in at the same time next week when we're going to be looking into space and out towards Mars, because we'll look at the next generation of Mars rovers set to venture across the red planet. The Naked Scientist comes to you from the Institute of Continuing Education at the University of Cambridge. We're supported by Rolls-Royce. I'm Chris Smith. Thank you very much for listening, and until next time, goodbye.